Okay, well, thank you very much for the few of you to have come back from the coffee earlier. So the subject of this presentation is about whether or not probiotics can help with AHPND, EMS, and white feces. This is a slide that has been shown recently by one of the major Thai company, CP, to show that after EMS, shrimp culture will never be the same again. And we had a nice presentation earlier by Dr. Soto. But some of the key points here I would like to emphasize is the fact that now you have many different strains of Vibrio parahermeticus involved, but also that the genes associated with EMS, HPND, are now being found in strains of other species. It was mentioned EMS in Australia. In Australia, it's not Vibrio parahermeticus, it's another species of Vibrio, which is associated with the EMS outbreak in Australia. Also, we now have reports of milder isolates in Mexico. They showed a very aggressive Vibrio that killed in a few hours. Some of the strains we have in Vietnam or Thailand are not as aggressive. And other are thought to be maybe linked to white feces. So not giving HPND, but something else. Finally, Flegel and other people in Thailand have reported other bacteria, other species, Shewonella and a few others, that could be associated with EMS, meaning not only Vibrio parahermeticus, but other species could work in synergy to lead to the outbreak. Actually, it was mentioned earlier, but I had prepared that to show the main problem is that bacteria exchange genetic material. They can do it by just incorporating genes from one species to the other, or they can do it by transferring this genetic material, which would be on a plasmid, which is the case of the Vibrio parahermeticus associated with EMS. So that means you have high probability of these genes being exchanged. And this is why it's not only screening for specific Vibrio parahermeticus, but more maybe overall the bacteria that could be associated with Vibrio strains. White feces, this is something that is giving problem in India, in Indonesia, in Thailand for many years. The cause is not so clear. In the past, it was associated with poor quality of feeds, mycotoxin and so on, but also has been associated with Vibrio. If we look in the literature over the last few years, you can find the list of Vibrio species which are associated with white feces in Indonesia, in India, in Thailand, and in China. So that means many of these countries are affected both by EMS, but also white feces, and Vibrio species are found in both cases. Okay, no need to look in detail for this. What it matters is to talk about the mechanism of communication of Vibrio, quorum sensing. This is important because it's the mechanism the Vibrio use to understand whether or not they reach a certain number, abundance of bacteria, and then they become pathogenic, virulent. What you can see here is that the same kind of molecules, signal molecules, are found in a large number of species of Vibrio. And these are all associated with disease and virulence. So, one more time, to control EMS, it's not only about controlling one or two strains of Vibrio parahermeticus. It's controlling much larger communities. So how do we cope with Vibrio? That means looking at solution both from broodstock, hatchery, nursery, and grow out. It's easier sometimes to just blame the supplier of the PLs or the hatchery blaming the broodstock supplier. Actually, we have to integrate all the steps. At the beginning of AHP and the EMS in Thailand, Flegel and his colleagues made a nice survey to show that the live food used in broodstock was a big part of the contamination. So at the end, the recommendations were reduce the use of live feed and screen the broodstock. Now in Thailand, they have also microsporidium EHP, so it's an extra step that is to be taken to screen the quality of the broodstock to make sure you don't transfer your pathogen from your broodstock to the hatchery and so on. If we look at the hatchery, there again, it's important to identify all the possible point of entry of the Vibrio. For example, the algal culture, there are some simple treatments, very easy, that can be done to control the Vibrio in the algal cultures. Same with the Artemia, to prevent the Artemia being a source of Vibrio for the PL, or for the myces, and also to control Vibrio biofilm. It has been mentioned also earlier, Vibrio, like most bacteria, create biofilm. Biofilm gives them more strength and the ability to resist against disinfection, antibiotic, and so on. So there are also methods that can be used to control the biofilm and therefore prevent the Vibrio from recolonizing other facilities. During the culture, now the use of probiotics to control Vibrio during the larva rearing is, I think, very well accepted. There's a lot of publication, peer-reviewed, to document how you can control the Vibrio in Zoea, myces, or PL, which allows you to make sure that when you transfer your PL from the hatchery to the nursery, you actually reduce the number of Vibrio and make sure you don't contaminate your nursery or your farm. Another step at the bottom part is to work on treatment. So you can work with some plant-based product to clean your PL, or you can work with some treatment with biocide that can also allow you to re-clean the animals before they are transferred. Finally, it's important again to look at the quality of the PL before they are transferred to the farm. This is something in Asia many people stopped when they started working with Vanamey. They looked at Vanamey as very easy shrimp, no need to check anything. We just get fast growing animals and we just produce. And they stopped looking at all this quality. With EMS, it came back. So now in Thailand, for example, the Department of Fisheries provides free screening. So the people can have their animal checked by recognized labs. Many also laboratories provide this service. That means you are sure your animal is checked. The new one now is, of course, EHP, which is a major problem now. But then also the standard analysis to make sure you have quality animals. Now, when we look in nursery, when EMS came, the interest in Asia was to study the concept of nursery, and basically Mexico. Unfortunately, nursery by itself was not a solution, as Mexico got hit by EMS. But what we have shown through working with companies in Mexico is that extra probiotic treatments to the normal protocol can give you an improvement on survival, but also on the growth of the animal. So if you select bacteria that can compete with the Vibrio, and you apply these in the right way in the nursery, you can really have an improvement on the growth and survival of the animal. Now, this is data from Dr. Put from the Department of Fisheries in Thailand. He presented it a few weeks ago in Thailand. It's very interesting because He looked at all samples from broodstock all the way to farm and looked for the plasmid of EMS and the toxin genes. And what they saw is that the farm water, the farm sediment, and the PL were the main source of contamination. So the treatment of the PL and juvenile, very important. That means screening and emphasis on quality in the hatchery. But then you need to look at the farm sediment. So when they observe that, the approach that can be taken is how can we control the sediment to avoid the Vibrio being dominant. First point, all Vibrio like chitin, the mold of the shrimp. So to remove this mold or to avoid the accumulation inside the pond is the first thing, to make sure you don't have support for the Vibrio. Second, Vibrio are very fast growers and they have the ability to stay in a state of not dormancy but slower activity. When there is nutrient, after algal crash or something else, you suddenly have nutrients coming and the Vibrio will be the first one to grow fast and colonize. So it's very important to avoid accumulation of the mold, but also keep the quality of the sediment to avoid having nutrients for the Vibrio. So you can see two ways. This is some of the new intensive system in Thailand. As you can see, it's a lot of piping, water exchange, flushing out continuously the waste, redesigning of the pond to concentrate the waste and flushing out non-stop. This is very expensive. It requires a lot of investment. If you are part of a big company, you can do that and it works. But for some, the medial player is much more challenging. And there are also issues that you produce a lot of waste that you release in the environment. So it's another point to consider. The other way is basically closer to what maybe is done in Latin America. It's looking at ways to control the quality of the soil with probiotics, but also, when required, do some flush. This is coming from super intensive in Indonesia. And as you can see, they have a central drain, they flush out, and the workers collect the mold and they spread the mold here. So they remove the mold and they observe if there are signs of disease, of mortality. So it gives them an idea if there is a beginning of an outbreak, and then they can do some treatment or modify the management. Now, how can probiotics help to control the environment? Probiotics can be selected for the production of enzymes. There are many enzymes. These are some of the enzymes that we focus on, and we work with the protease, amylase, cellulase, and so on, because they can help to degrade algal material, but also uneaten feed ingredient or dead animals, and the feces. So it's a tool to allow you to degrade the waste. It's also important to explain how this is done. So when we register product, we need to go to an external lab. This is the method they use, the substrate used for the test, and it means we can explain how we can claim, why we claim we have these enzymes and how they work and the activity. This can be done with all registered, accredited laboratories. Once we select probiotics to degrade the waste, the consequence is that we want to have less waste in the pond. If we have less waste in the pond, as was mentioned also earlier, less nutrient for the Vibrio to grow, lower probability of anaerobic conditions, less stress for the shrimp, and also lower production of ammonia sulfide and so on. So less Vibrio, less chance of Vibrio growing, and also less stress for the shrimp. These are data from Brazil to show that if you apply probiotics in the water and in the feed, you can remove the Vibrio from the sediment. In this case, this is something we did in 2007. So we've been working on it now for over 12 years. We have data worldwide. And we can show that the bacillus, in this case, can be recovered from the pond bottom, because this is where everything happens, while the Vibrio can be displaced. It was important because when people looked at that first, they were focusing on the water, and they could not see any trend. And they said, your product don't work. I check the water, no difference. But bacteria go where there is substrate, where there is nutrients. It's in the pond bottom. So it has to be effective in the sediment. Now, when we look at white feces or EMS, it's important to include the shrimp gut treatment. So in this work here in Thailand, they looked at Vibrio havei and how it could be recovered from different parts of the gut and be associated with the disease. EMS, whether or not it colonizes the stomach or it just accumulates there, it's another area where we need to find a way to control the Vibrio from the gut of the shrimp. How do people do that? First, you can select probiotics that can kill the Vibrio. So in this case, this is a strain of Bacillus. These are Vibrio parahemolyticus from a culture collection. So you can buy them and test yourself. And it shows you inhibition. This is Vibrio havei, and you can do a different method to show that your Bacillus kill directly the Vibrio. The second point, which maybe is more important, is how the probiotics, when they are put in the feed, can displace the Vibrio from the gut. It's work that we did also almost 8-9 years ago. And it shows you the Vibrio in the gut of the shrimp, yellow or green colonies, and then you start to put the probiotics. No more, you stop, it comes back. It was done in Australia. The message is, if you can occupy the gut with the Bacillus, you will have less chance of the Vibrio being present. So you can reduce the Vibrio by just occupying. This was done with traditional method. Now we work with more high-tech techniques. This is work done by a team in Brazil, a PhD student from FURC in Brazil, colleague of Doriano, and she worked on seeing how probiotics can reduce Vibrio in the gut of shrimp, whether you work with biofloc or traditional. And what it shows you is that if in biofloc you put the Bacillus in the water, you recover them from the gut of shrimp. It's normal. In clear water, you recover them also. And at the same time, if you look at the Vibrio, if you put the probiotics, you reduce the number of Vibrio in the gut. If you go clear water, you reduce. So the message is, it's possible, whether you work in biofloc or in clear water, to reduce the number of Vibrio from the gut of the shrimp. So we can do it in the sediment, but we can also do it inside the shrimp. You saw a graph of challenge test earlier. Is another one. This one is done in Vietnam. In this case, we provide the probiotics in the feed, and then we challenge the animal. This is the mortality of the shrimp with the EMS. In this case, for the challenge test, it should target 50% mortality. This is the goal. So we adapt the dosage of Vibrio to reach 50% mortality. Then the next step here, you do nothing, no challenge, no mortality. In green, it's antibiotic in the feed. And you can see you can improve by reducing mortality. In here, this is the probiotics in the feed. And you can have even a further improvement. So it's logical. If you remove the Vibrio from the gut, you have less chance of this Vibrio reaching a large number enough to create the disease. Other benefit that you can see from probiotics in the feed is they will reduce the FCR. What it means is, okay, lower feed cost, which is good, but also less waste in the pond bottom, since the food is taken up instead of being wasted. So one more time, less chance of Vibrio growing. And second, the bacillus will stay in the shrimp feces. So these feces are important because the shrimp eat their own feces. So if you colonize the feces, the Vibrio, which are fast grower, will have less chance of dominating the feces. You have less waste, and again, less chance of Vibrio. So it's additional ways in which we can control. So if I reach some conclusion for this, the reason probiotics can help for EMS or white feces, the first one is controlling the waste, so less chance of Vibrio growing. They can kill directly the pathogen, but they can also improve the feed utilization and therefore less waste in the pond bottom. So one more time, reducing the growth of Vibrio. And what I showed you, they can occupy the environment, whether it is the pond sediment or the shrimp gut. So these are all angles that allow us to reduce the Vibrio. It's not to remove them, it's just to maintain the Vibrio at an acceptable level. But we have to insist, it's not the magical bullet. Probiotics cannot solve everything. It's just one tool for the farmer. And that means, this is why I emphasized at the beginning, it has to go from the broodstock, hatchery, nursery, and so on. And prevention of transfer from each step. So it's only through this holistic approach that disease like EMS or white feces can be handled, and that the probiotics can be part of it. Thank you very much. Muchas gracias.
Thank you, Olivier, for your presentation. This is for you. Since all of you had a really good time, ya que él